Hello everyone, I am Dr. Richa from Physiology Open, and in this video we are going to continue our series of pharmacokinetics. So today we will talk about metabolism of drugs. So what is this metabolism of drugs? Actually, in metabolism, the drugs are biotransformed. So this word you will see, biotransformation, meaning what? That means they are changed biologically. They are transformed biologically. Basically, there is some chemical changes in the drugs. And where does this biological transformation occur? Well, the maximum transformation occurs in the liver, but it can also occur at other organs like intestine, lungs, and even in plasma. Kidney we are not talking here because kidney it is mainly excretion. This biotransformation, chemical change in the drug is occurring. And why it is occurring? Well, first of all, it is a foreign substance and body deals with foreign substance in its own ways. However, with drug, there are various outcomes which happen with the chemical transformation of the drug, and ultimate target is to conversion of lipid soluble substance, lipid soluble drug to lipid insoluble, or we can say water soluble substance, so that finally it is excreted. Okay, but we have different outcomes for this drug. One is that active drug which has been administered, it will be inactivated by this metabolism. Then there might be active drug which then after metabolism becomes active metabolite. So final action of the drug will depend on a combination of both this, that how much active drug is present and how much active metabolite is present. And then there is a prodrug which we administer as a prodrug, and then as it enters into the liver it is activated. Okay, so this helps us in changing the bioavailability of the drug. Say suppose there is some drug which is inactivated in the stomach, so it might be administered as a prodrug, so that only when it is absorbed in liver it is converted into a active component, then only its action is going to start. Okay, then because of this conversion there might be certain drugs which can form toxic metabolite. Okay, for example, paracetamol is there, which in the liver it is converted into N-acetyl benzoquinone imine. That component is formed which is toxic to the body. Okay, so that can also happen. Now, how this metabolism is occurring? Well, there are two ways. There are phase 1 reactions and phase 2 reactions, but remember that ultimately the target is to convert lipid soluble drugs into water soluble drugs. Okay, so the drug can undergo either phase 1 reaction, okay, or it can undergo phase 2 reaction directly, or it can undergo first phase 1 reaction and then it can undergo phase 2 reaction. So suppose with phase 1 reaction it has become water soluble, but that is not enough for excretion, then that drug will undergo phase 2 reaction. So what are these phase 1 and phase 2 reactions? Phase 1 reaction is also known as functionalization or non-synthetic reaction, and phase 2 reactions are also known as conjugate or synthetic reactions. Now in phase 1, we have 5 different types of reactions, that is oxidation, reduction, hydrolysis, cyclization and decyclization. Okay, so oxidation, that means the addition of oxygen, or we can say removal of hydrogen. Okay, reduction, where there is the addition of a hydrogen. Hydrolysis, that means a water component is added. Cyclization, the composition of the drug is changed so that it can form a ring. Okay, and decyclization, that means the original drug was in the form of a ring and now it has been opened. So these are functionalization or non-synthetic reactions. Phase 2 reactions are conjugate or synthetic reactions. That means a separate component is added to that is known as conjugate. So a separate component is added to this and that forms some other products. Okay, now these reactions, phase 2 reactions, are brought about by enzymes known as transferases. Okay, because they are going to transfer a component to these drugs. Okay, for example, say suppose this is a drug and these are the various substances to which it is conjugated. Okay, so drug can be conjugated to glucuronide, to glycine, glutathione. It can be conjugated to acetyl group, methyl group, sulfate group or nucleoside group. So total we have 7 reaction types in this. Okay, so conjugate reactions can occur with 3 Gs, that is combination with glucuronide, glycine, glutathione, or it can be acetyl group, methyl group, sulfate group and nucleoside group. Now, after phase 2 reaction, definitely the drug becomes water soluble, and that after this it is inactivated and it is excreted. However, there are certain exceptions to this. One is morphine, okay, morphine, that after glucuronidation it becomes active, and second is minoxidil, where it is sulfated, okay, so there is sulfate conjugate of minoxidil which becomes active. So these are exceptions. Mostly it becomes inactivated and is excreted. Now, certain things about it. First thing is that the most common reactions among this is oxidation reaction. Second most common in this phase 1 reactions is the reduction reaction. Among the phase 2 reactions, most common is glucuronidation. Okay. Now, these reactions are brought about by certain enzymes. There are various enzymes in the liver, and these reactions are brought about by certain enzymes. As I told you that conjugate reactions, we have transferases. So, how to name these enzymes? So, we will have, for glucuronide, we have UDP, uridine diphosphate, glucuronyl transferase. Okay, this is the same enzyme which you have read in bilirubin metabolism in physiology. Okay, so UDP, glucuronyl transferase is there. Then we have glycine transferase, glutathione transferase, then N-acetyl transferase, methyl transferase, sulfate group transferase and nucleoside transferase. So these are the various transferases. Now, these enzyme activity varies from person to person. Understanding. So these reactions will also vary from person to person. That means the inactivation of the drug, or these outcomes which we are talking, these outcomes can occur with the drug. Mostly these outcomes occur with phase 1 reaction. Phase 2 reaction, mainly it is active to inactive component. Okay. So these reactions will also vary from person to person. That means the drug activity will be different from person to person. We may see that certain drugs may not act that much well in certain people, because if you are given a pro-drug and it is activated, maybe in that particular person that enzyme is not that much expressed, maybe the quantity is not that much, so the active metabolite will not form that much, right? So these are the various reactions. Fundamentally, this variation in the enzymes, that is known as polymorphism, genetic polymorphism, right? Now, for oxidation, one very important enzyme is CYP450. And this is very important because this concept you will see again and again in different chapters, that there are various enzymes which are acted upon by this enzyme. So that is known as substrates. Substrates are the drugs which basically this enzyme acts upon. Okay, that is substrate. But there are certain drugs which are its inducer and inhibitor. That means some drugs will increase the activity of this enzyme. Those drugs are known as inducers. Some drugs will decrease the activity of this enzyme, and those are known as inhibitors. So what are this CYP450 enzyme? Well, the name is cytochrome. This is CY means cytochrome. P is for pigment. Okay, and 450 is the wavelength of the light in nanometers. Okay, CY is cytochrome, P for pigment, 450 is the wavelength of the light. Actually, these enzymes are of different types. First thing is that some enzymes, all the enzymes which we covered, some enzymes can be microsomal and non-microsomal. Microsomal means they are present on the endoplasmic reticulum, while non-microsomal can be present in the cytoplasm or in the mitochondria. Okay, most of the enzymes, these are microsomal. Whatever we have discussed in oxidation enzymes, those are microsomal. So these cytochrome P450 are microsomal, and glucuronidation also, these are the microsomal enzymes. Now, among this also we have a pigmented enzymes. Okay, so that is what this is. This is a group of heme proteins, and heme is what? Heme is a pigment, right? So that is why P for pigment. Now, if this enzyme is taken out, suppose the cells, liver cells are taken out and they are broken in a test tube, and this enzyme will be released. Now, if we measure this in a colorimeter or a spectrophotometer, actually we pass a light. So what happens that these enzymes can absorb light at 450 nanometer. So that is why the name cytochrome P450. Okay, but when we refer to these enzymes separately, you see here I have referred so many enzymes, nowhere I have written 450. So this word is generally understood and it is not repeated when we are writing the names of these enzymes. So there are so many of these enzymes, many enzymes are there, and basically they have 3 families. Okay, so that's what is written, 1, 2, 3 families of cytochrome P450 enzyme. Among these families we have various subfamilies. Subfamilies are denoted as A, B, C. Okay, family is denoted as 1, 2, 3. Subfamily is denoted as A, B, C. And in these subfamilies we have various isoforms also, and this is denoted as 1 to 19. Okay. So this subfamily is not limited to ABC. There are others as well. D, E also is there. But isoforms, they are 1 to 19. Okay. So whenever we refer to these enzymes, you see these number here. So if it is written CYP3A4, what does it mean? This is cytochrome enzyme. P is for the pigment P450. 3 is a family, A is a subfamily, and 4 is the isoform of the enzyme. Okay. So there are a number of these enzymes. Many enzymes are there. However, for us, the significant ones are the 6 enzymes. Okay, so these 6 enzymes I have written, that is CYP3A4, CYP2D6, CYP2C9, then you add one here, CYP2C19, then CYP1A2 and CYP2E1. Okay, 1A2 and 2E1. Now, these enzymes basically metabolize the maximum amount of drugs, and among these also, these two, CYP3A4 and CYP2D6, are the ones which metabolize the maximum amount of the drugs. So what is this known as? These drugs which are metabolized by these enzymes, these are the substrates. Okay, so 50% of the drugs are metabolized by these 2 enzymes, rest we have different drugs. So these names you are going to see again and again in different chapters of pharmacology, right? Now there are some inducers and inhibitors, as I already told you. The most common ones I have mentioned here, but again you will see some other drugs as well, right? So we have rifampicin, which is an antitubercular drug, which is an inducer of all these enzymes except CYP2E1. It induces all these enzymes. That means it increases the activity of these enzymes. Then we have barbiturates. Barbiturates also induce all these enzymes except the last 2 ones. Okay. Then nicotine. Nicotine causes the induction of CYP1A2 enzyme, and alcohol finally causes the induction of CYP2E1 enzyme. Okay. So these are the various inducers. We have phenytoin and CBZ, basically, which are anti-epileptic drugs, which are inducers of CYP3A4. Apart from this, we have glucocorticoids and isoniazid. Isoniazid is also an antitubercular drug. Okay, so rifampicin and isoniazid are antitubercular drugs. Then we have anti-epileptics, which are the inducers. We have barbiturates, then smoking and alcohol. Okay, now if you don't remember it right now because everything is new for you, don't worry, revisit this video or revisit this chapter again when you are reading about the other drugs. So then it will help you consolidate your concepts. Okay, the inhibitors of this enzyme include erythromycin, ketoconazole, fluconazole. In fact, you will see that inducers are lesser. There are so many inhibitors of this cytochrome P450 enzymes. Okay, then one more concept you remember, that induction of this enzyme takes little time. Okay, it may take from around 1 week to 2 weeks for this effect to take place. However, inhibition is faster. Why? Because for induction, actually sometimes you need to synthesize, the body needs to synthesize the enzyme as well. So that needs time. However, inhibition means that whatever active enzyme is there, that is inhibited. Okay. So the effects which we will see in the activity of the drugs, that will be taking time in case we are seeing the effect of the inducers. On the other hand, the inhibitors will act faster. Okay. Now, why do we need to know about this enzyme inducers, enzyme inhibitors? What are the substrates? Well, there are so many drug interactions which can occur. Okay, so we'll talk about certain examples in this as well. So suppose there is one drug which is a substrate for this enzyme. Okay, so this one is the substrate where this we saw the outcomes that is going to take place. But there is a separate drug which induces the activity of this enzyme. So what is going to happen? Obviously this enzyme is going to become more active and there will be increased metabolism of this D1 drug. So this can lead to therapeutic failure. That means you have given the drug, it is metabolized so fast, then it will not be able to act. Okay, one example of this is giving OC pills along with rifampicin. So rifampicin, I told you, it is anti-tubercular drug, and OC pills are given for contraception. So if OC pills are given with rifampicin, then the failure of action of OC pills can occur because it will be metabolized very fast and ultimately it can result in pregnancy. The therapeutic failure of OC pills can occur. Then it can also lead to toxicity. Say suppose there is consumption of alcohol and the person takes a paracetamol. So in that case, alcohol is going to induce the enzyme which is responsible for the metabolism of paracetamol, and I told you that paracetamol toxic metabolite is produced. So this can lead to toxicity as well. The accumulation of the toxic metabolite will be there, right? Then second example, say suppose there is one drug and now this drug induces its own metabolism. So this drug is the substrate also and it is the inducer of the enzyme also, same enzyme which is the basically the substrate also same and the inducer also the same. So what is going to happen? Well, the same drug will be metabolized much faster. So it will lead to tolerance. That means over time the action of the drug will decrease, and that happens in case of carbamazepine. It is the inducer of the enzyme for which it is the substrate, and also chronic alcoholism because for alcohol this same enzyme is the enzyme which metabolizes it, and this same enzyme is induced by alcohol. So this leads to alcohol tolerance as well. Third, suppose there is a drug and it inhibits its own enzyme. Okay, so what it is going to occur? The metabolism of the drug is going to decrease. That can lead to increased bioavailability of the drug. Yes, because the drug is not being metabolized. Suppose this enzyme is making this drug inactive. So now that is not going to happen. More active drug will be in the circulation, and this happens in case of verapamil and omeprazole. Okay, so verapamil inhibits its own metabolism, and this can lead to increase in the availability of the drug. It can lead to increase in the half-life of the drug in the circulation. Then fourth is like there is a drug and there is another drug which inhibits the enzyme. So this is opposite of the first one. Okay, these two are auto induction and auto inhibition. Okay, so here it is inducing its own enzyme and here it is inhibiting its own enzyme. In the first one we saw there is separate drug. In the fourth one also there is a separate drug. So this is inhibiting the enzyme. So what is going to happen? Again, the drug is not being metabolized because the enzyme is going to inhibit it. This is going to increase the bioavailability of the drug. So one example of this is like giving protease inhibitor, which is for HIV, given for HIV, with another drug, ritonavir. Ritonavir is the drug which inhibits the enzyme. Okay, so this is used therapeutically for increasing the bioavailability of this protease inhibitor in the body. Okay, then it is used also for endogenous substances. Say suppose endogenous substances are there, for example bilirubin. Bilirubin is metabolized by one enzyme, and if we give another drug which induces this enzyme, then what will happen? This endogenous substances, there will be increase in the metabolism. In fact, this is used therapeutically for congenital non-hemolytic jaundice. There is a syndrome, Gilbert syndrome, in which there is decreased activity of this enzyme which we saw. Okay, I told you this is used for the metabolism of bilirubin. So that means bilirubin will accumulate in the body. If we give a drug, say suppose phenobarbital, that is the barbiturate. Barbiturate is the inducer, isn't it? So this phenobarbital also induces this enzyme, and this in turn leads to increased metabolism of the bilirubin. So it can be used therapeutically. Similarly, in Cushing syndrome, if we give phenobarbital, that is also going to increase the metabolism of this cortisol. Okay. Then if we have liver diseases, what is going to happen? You know, all these enzymes, main organ is liver. So if liver disease is there, then the metabolism of the drugs will be affected. Hence, their active concentration will circulate in the body for longer duration, and it can lead to toxicity as well. So we have to change the dosing of the drug if there is liver disease. Okay. Then one more is there. Say suppose there is a drug drug 1, which basically is metabolized by one enzyme, and there is another drug which is also metabolized by the same enzyme, then there can be competition between these two drugs that which will be metabolized. Okay, so if one is metabolized more with this enzyme, then the other drug will not be metabolized and it will accumulate in the body. Okay, so those were the fundamentals about the biotransformation. Briefly we saw where it is occurring, why it is occurring, how it is occurring, two reactions, phase 1 and phase 2. In phase 1 we saw there are 5 reactions, in phase 2 we saw there are 7 reactions. These are brought about by transferases. Then the enzymes which are there, there is genetic polymorphism, because of which the activity of the drug in different people can vary. Then we saw about the CYP450 enzyme, why it has such a name, what are the families, subfamilies, isoforms, what is the difference between microsomal and non-microsomal enzymes. Then we saw that various CYP450 enzymes, mainly the 6 ones, and what are the inducers and inhibitors of these enzymes, and then we saw the various drug interactions as well. Thanks for watching the video. If you liked it, do press the like button, do share the video with your friends, and don't forget to subscribe to the channel Physiology Open. Thank you.